Hey everyone, it's Mrs. Shannon. And I think we are going now, so I want us to keep talking. I didn't know we were on. But this is David Nufke's Prezi on cellular energetics. And this is the third one. It's going to focus on regulation. It's going to focus a lot on the enzymes, which is what number one left off with. I'm sort of skipping number two for now, but I'll come back. You'll get a number two at a later point. It's on thermoregulation and sort of different mechanisms that organisms have evolved over time in order to regulate metabolism and the use of energy. So the second Prezi focuses on that sort of evolutionary aspect. This Prezi is going to focus more on the organismal aspect and how this energy is regulated within an organism or within a reaction, for instance. And so we're going to go from the theory to the practice. And so the big questions here are, how do living systems control their metabolism? We know what metabolism is at this point. We know which type of reactions will proceed. We know which type of reactions need to be coupled together, for instance. But now we need to know how it gets balanced in an actual living system. And that's what this one is all about. How do living systems carry out variety of specific chemical reactions? How do they know when to have them go and when to have them stop? And how does everything kind of dance together? Okay, enzymes are the big regulators, the big kahuna, that's the word I'm looking for, the big kahuna of the energy world. They're just biological catalysts. Most enzymes that we encounter are proteins. Although there are some RNA molecules that can function as an enzyme too. And we actually encountered that when we were looking at some of the origin of life theories and the whole idea of RNA being probably the forerunner of the type of organic molecule that would have transmitted information, because it could also act as a molecule that could be an enzyme. So it could be dual function. So if you remember the origin of life readings that we did, they talked a little bit about RNA molecules being used as enzymes. You know an enzyme when you're reading because it ends in "ase." It's a common nomenclature suffix for enzymes. When we talk about it, we usually refer to the enzyme substrate. So whatever the enzyme is working on. So when we did toothpick case in the class to model, our fingers were the enzyme, but the substrate was the toothpick. And so we called it toothpick case. Our fingers became known as toothpick case. So the enzyme ends in "ase, and it usually refers to the name usually has something to do with a substrate in it. So how do enzymes actually work? Well, they interact with the substrate. The substrate is sort of what either one or multiple molecules that are going to be either split apart or put together. And so here you have an example. We have an enzyme, and then we have our reactants right here. So in this case, in this picture, we have two reactants. These make up our substrate. So when our enzyme and our reactants come together, we have an enzyme substrate complex. Remember, a complex is any combined molecules. Enzyme substrate complex. During the course of this, the reactants are brought close enough together that they can rearrange, products are formed, and then they are released. The work is localized to an area of the enzyme called the active site. And so right here, where the reactants actually bond, where the enzyme and the substrate actually meet, that is called the active site. So this part back here doesn't do much. This part is where all the action is happening. So that's the active site. Now, we have what's called an induced fit. The shape of the active site of an enzyme is shape specific for a particular substrate. So in this case, this will perfectly fit in this active site of this enzyme. The binding of a substrate to the active site induces any kind of conformational change needed to catalyze it. And so when the substrate locks in here, when it gets to that spot, if any kind of conformational change is needed to start the reaction, it will happen. And so you will actually see here when the complex is formed, when the substrate hits that active site, that it does close down a little bit and there is a conformational change here. Examples, topoisomerase. It's involved in minimizing mechanical stress of DNA during replication. It's kind of like a nurse enzyme. It makes a temporary cut in the helix to allow the reaction to proceed. Rubisco is another one. It attaches carbon dioxide to sugar. It's a precursor molecule in photosynthesis. Okay. And so here you can actually see the DNA double helix, and you can see right here the enzyme. Okay. 50% of all protein found in the chloroplast is rubisco. And so here you can see the enzyme substrate complex. Most enzymes require like these accessory compounds, vitamins and minerals to help it become active in an active state. And it does it by changing its form a little bit. This helps regulate whether or not enzymes are on or off. And so they call them cofactors. Now, some of the reasons for this is evolution. Evolution plays a central role in enzyme structure and function. Various studies have been conducted to investigate the effect of evolution on enzymes. And what they notice is that this regulation piece has sort of separated changes that have been successful from changes that haven't. And some of these studies that they've used to support this idea is analysis of enzyme genes. So you're looking at genomic studies, looking at the DNA. They will actually do artificial selection of enzyme activities in laboratory settings to see how they perform. So we're looking at sequencing, we're looking at actual lab studies, and then looking at an ethnographic, demographic studies of enzyme genotypes and how they determine phenotypes. That sounds complicated, but basically looking at the prevalence of different enzymes in different populations and seeing how they correspond to different phenotypes that they see. Example would be like lactose intolerance and the presence of the lactase enzyme. So there's a few different ways that you can go, that researchers go about looking at the impact of enzymes in evolution. Now, enzyme function can be stimulated or inhibited by factors in the cell itself too. And we have competitive and non-competitive interaction. Competitive interactions are anything that competes for this active site, any other molecules that compete for this active site. And so here we have a competitive inhibitor. It's one that gets in the way of the active site and the substrate is no longer able to bind. A non-competitive inhibitor interaction is one that will have a molecule bond to it, an interaction occur, or an inhibitor come up. You'll see it called both. And it changes the conformation, or it changes something about the enzyme, but it doesn't actually take its spot on the active site. Like the substrate itself could still come into this, but at the same time, it doesn't fit anymore. So the active site is still available. Nothing's being done to that. Nothing's taking the spot, but the shape itself in this case is changing, or something else about the environment is going to make it unfavorable for this connection to form. We have what's called allosteric interactions. These are other site interactions. They stimulate or inhibit enzyme activity by causing a conformational change. And so if we were looking up here, this non-competitive inhibition, it's an example of an allosteric interaction. So when something binds somewhere else, changes the shape of it, and it's no longer on. So here we have an allosteric interaction that activates an enzyme, where you actually need a molecule to get it to go. Okay, on the flip side, we have an inhibitory one. Binding of an inhibitor molecule can stabilize the enzyme in an inactive conformation so it doesn't proceed. So here, when the activator binds, shape changes so that it can proceed on. It's turned on, it's ready to go. Here, it's turned off, it won't proceed. And so it's like a feedback on-off switch. Binding of a substrate molecule to an active subunit of an enzyme can also trigger stabilization of the active conformation in all subunits. And so oftentimes there's not just one enzyme in the complex, you have multiple ones. So in this case, you have a complex of 4 caspase. We have different subunits, different protein domains that come together. All we need is one allosteric interaction on one of those subunits. It signals to the other ones to change the shape as well. That gives you 3 binding sites. So in this particular case, yes, one site is going to be occupied by this allosteric regulator, but we still have 3 binding sites now on, whereas before we had no binding sites. Example is caspase. It's important enzymes for cell death, also known as apoptosis. There's been data from an experiment to determine if caspase enzymes can have an allosteric site. The active and inactive forms of caspase 1 were already well known. The hypothesis is that allosteric inhibition of caspase 1 will lock the enzyme in an inactive conformation. So basically, they were trying to see if this would lock up, if they could inhibit from caspase 1 from turning on and signaling apoptosis. Whoopsie. And so here we go. We have the active form right here. Add an inhibitor. It's now the inactive form. It's not going to work. And you can see the whole conformational change here. You can see the multiple domains here. Okay. Here in caspase 1, the substrate's able to bind to the active site. We're good to go. However, with an allosteric inhibitor changing the shape, it is no longer able to bind to the substrate. That's kind of like looking at the group with toothpick ace that did the oven mitt example. Something important to know, and it goes back to what we talked about with the important need of compartmentalization in cells, is that localization of specific enzymes and any of the reactions they mediate within compartments allows for control of reactions to occur. Because it is contained, it's not going to interfere or damage with other things, other reactions, other products being made in other parts of the cell. There's environmental influences that do affect the rate of a reaction. Like all proteins, enzyme structure and therefore the function can be affected by the condition of the environment. There are 3 major environmental conditions that affect enzyme structure and function. This you should take note of right here. These are very characteristic of the 3 factors, environmental factors that will affect it. First one is we have temperature. We have a prime temperature above which we have denaturation until it goes, until the reaction stops completely because the change of the shape is so dramatic that it won't function. It usually has a longer curve. It can go much lower than it can higher usually. This slows down the reaction. It slows it down less occurrences of it interacting because it's molecularly moving slower. Here it's actually going to be changing the shape. So there's always going to be an optimum. pH, again, an optimum, above which denaturation, below which denaturation. Concentration, when we're looking at enzyme, substrate, and cofactors, they're limiting. It's kind of like a carrying capacity for a population, limited resources, they reach a carrying capacity. Well, here we have like a reaction capacity. we are limited by the presence of enzymes, substrates, or cofactors. So it's not going to go on indefinitely. Either all of our enzymes will be bound up with substrates, or we run out of substrate, et cetera, et cetera. Many metabolic pathways have regulatory effects on enzymes that catalyze it. We haven't talked a whole lot about feedback loops. So we're going to be talking about that in the next unit. But basically, it's sort of on-off checks as we go through. Signal checks to see if a pathway should continue to occur or if it should stop, if products should be made or if it shouldn't. And so in this case, we have our initial substrate. We have it bind to an enzyme. We have different intermediates that are acted upon by other enzymes until a product is formed. However, if we have the presence of certain molecules in the area, for instance, in this case, isoleucine, we have it bind allosterically. It acts to inhibit the reaction because it's changed the site. It will no longer proceed this direction. And so what this does is it keeps too much product from building up. So if we start with threonine and we're trying to build isoleucine, if the enzyme has a spot to recognize isoleucine in it, to recognize the presence of it as it builds up, the isoleucine, as it builds up in the area and comes into contact with the enzyme, it's going to turn these enzymes off. So it will slow down the actual production of it. So from this Prezi, make sure that you can explain how enzymes function as catalysts, how they lower that activation energy. Explain the induced fit model of enzyme function, how once that enzyme and substrate combine at the active site, how the enzyme is then turned on. Provide examples of enzyme catalyzed reactions in biological systems. Explain the relationship between enzyme structure and function, and explain the major modes of regulation of enzyme activity. And so when they're talking about major modes of regulation, think about that allosteric regulation. Think about inhibition. Think about competition. Think about regulation, environmental regulation with pH and temperature. Think about feedback regulation with the products and the enzyme working allosterically to regulate the binding to the reactant. So those are just some of the ideas of how enzymes can be regulated. So let me know if you still have questions, and I'll talk to you on the next Prezi podcast.